So today I'm going to show you how the first day of Lab 1 for microbiology was performed. So in this one part, what we've done is we've taken an equal amount of S. marcescens, Serratia marcescens, and Escherichia coli. Both of these are purchased from Carolina so that we know that they're super pure. And we've made a mixed culture. This is a mixed culture, a 1-to-1 mix. So we know that there's equal parts of E. coli and S. marcescens here. We know that this is mixed and not contaminated because we know what both of the species are. So now what we're going to do is we're going to take this culture and we're going to do the quadrant streak method on that. So in the quadrant streak method, we are going to take our loop, and I've already got my flame going, as you can see here. Oh, home movies. Deal with it. Sorry. We've got our flame going right here. And of course I'm wearing my lab coat and gloves and goggles and all of that. Hair tied back, closed-toed shoes, all of the appropriate PPE. And what we're going to do is we're going to first heat the loop, as much of this turning bright orange as possible. So going all the way down. And this is an aluminum and nichrome wire, so it's not going to stay hot for very long. But of course you would know if we took this loop, which was just in that flame and became red hot all the way from the tip down the shaft, that this would burn our skin and it would certainly kill any of those microorganisms that we're trying to pick up. So that's against the purpose. So we're just going to let it sit for about 20 seconds, which is what we're doing as I'm explaining. Okay, and of course, we'll take this opportunity to explain about Sterile Technique, which I started before this video started. So the first step of Sterile Technique is to turn on the flame, which is a little counterintuitive. And then you take your bench cleaner, today I'm using ethanol, and then you'll, I'm going to scoot those out of the way, you'll clean off your counter. Lots and lots of people want to clean the counter first. But really, what happens there is that yeast and dust and all sorts of nasty stuff in the air will fall back onto the counter if you don't have this flame going. So what the flame is doing is this is directing the air up and out. So it's, instead of things falling down onto the surface, they're going up and out. So the dust is going up and away from our nice clean work surface. So flame first, counter second. And then you take your flame and you make that perfectly sterile. So now I feel like we've been talking enough time that this is sufficiently cooled, and we're going to take our 1-to-1 mixture and being careful to not touch the inside of the cap, because this is where you see the cap? This is where this one back here. This is the cap. Don't touch the inside of it. Any part that's touching the inside of this is contaminated with, not contaminated, but it's got our mixed culture in it. So if we touched it, we would contaminate it. So we want to treat that as perfectly sterile. So anything that's inside of this cap and inside there, we're not touching if we don't have to. This loop is now sterile because we sterilized it in the flame, made it red hot, that killed anything that was on it. So this is okay to put on there, but we still don't want to, just in case we didn't do a great job, we don't want to touch the sides if we don't have to. So we're just going to dip down and do a gentle twirl. And what this has done now is this has picked up a small amount of liquid, which I hope you can see right there. That liquid is a 1-to-1 mixture of E. coli and S. marcescens. So now we're going to take our plate, which is also sterile, and we're going to keep the lid down so that the clean part, the inside part, is facing up temporarily. And we're going to take this and we're going to tap, tap, tap and get that liquid off the loop and onto the plate. From the plate, now we're going to swipe, 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 back and forth. You want to keep your loop at an angle, not perpendicular. Perpendicular will stab into the agar, the solid medium here. This is LB agar. This is a rich medium. It will grow anything. And as you can see, it's solid. And we're just going to gently, 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 this is legitimately like drawing on Jell-O. Just gently run it across so we get like a nice, see this part is cloudy from here up. So we've just gently streak that all the way across. So that's our first quadrant. Now we are going to make a dilution. And the way we're going to take a dilution is that there's a whole bunch of bacteria on the loop right now. So we're going to sterilize the loop again and get that bacteria count back down to 0. And we're going to let it sit. So we sterilized that in the flame. Now we're letting it sit, and there you can really see the first quadrant there. And we're going to let this sit for 20 seconds. And just to make sure that this is really, really, really the same temperature as the agar, you can put it into the side. So you can stab it directly into the side there. I don't know if you can see it, so it's like straight up against. Okay, so now this is definitely the same temperature as the agar plate. I'm going to scoot this flame a little bit out of the way. And now we're going to do the second quadrant. So there's several different ways you can do the second quadrant, but it all starts by, as of the origin, the first quadrant is the origin for the second quadrant. So you're going to put your loop, not perpendicular, but maybe at like a 30-ish degree angle, and gently start writing, start streaking, and you pick up at the end, and you put back down, making all these lines, about 6, 7 lines, whatever you can fit, and that's how I make my second quadrant. I don't know if you can see it, but there you see those lines that go straight across. They all originate here in the first quadrant. So what I'm doing is I'm picking up some of those bacteria, but not all of them, and just making a nice line. So hopefully these bacteria, just a few of them, will now grow along these lines that I've made. So that's our second quadrant. So it's going to have fewer than our first quadrant because we made a dilution. So now back. We're going to flame this again. Make sure the tip is red hot. And flaming, flaming, flaming all the way up the shaft, because we want to make sure to limit as much contamination as possible. So there's our loop again. And now this time, there you can see the lines going this way. Move this out of the way maybe. Maybe you can see it with here. So you can see we've done the first and second quadrants. And now we're going to do the third quadrant. So again, waiting 20 seconds and then double checking. You can put the loop back into the side of the agar just to make sure that it's nice and cool. And then we use the second quadrant lines to make the third quadrant lines. So just straight across there, just making a few lines. Okay. And so there's our third quadrant, and again we're going to flame in between, and then we're going to make our fourth and final quadrant. So the goal of this is that we are going to see individual colonies. And the purpose of looking at individual colonies is that each individual bacterium looks a little different. So S. marcescens is actually pink. So for my fourth quadrant I'd use one line, and then I just use one line and I go choo 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 choo. So it kind of looks like that. You can see that. And then when we're all done, we cap this. So S. marcescens is going to be pink. So there's our final product. You can really see the first quadrant is now here, second quadrant is here, third quadrant is here, and fourth quadrant is right there. And with these you can see a little bit better. Okay, so the purpose is hopefully we'll have single colonies either in this third quadrant or this fourth quadrant. And then from there, we'll know that S. marcescens is pink and E. coli is white. So from there, we're going to take this plate, which will be provided for you in lab, and you're going to sketch this plate in your results, because you're taking this plate and you're going to isolate S. marcescens and E. coli, because on agar, they look very, very, very different. In liquid form, I don't know if you can see this, but they pretty much, you wouldn't be able to pull out an individual colony from here, or an individual colony from here. So liquid is really great for growing a whole bunch of bacteria, but solid is really great for separating bacteria, especially of a mixed culture, and making sure everything's growing great. So I'm going to be right back with an example of this. So here's a quadrant method that was done yesterday. And this is for Bacillus subtilis. So we weren't sure if this Bacillus subtilis was contaminated or not, so we did a quadrant streak on it. So the first quadrant is here, first quadrant is here where it's the most growth, and then here's the second quadrant, third quadrant, and then see in the fourth quadrant we have just these individual colonies. So these individual little circles, those are individual colonies, and that's what we're looking for here. So you can open up this plate when you have a nice sterile field going with the flame, which we still have. And you can see that these are very distinct. So these have, these are spore forming. They don't have like they're not like perfectly circular. You see that they're, it's hard to focus on it, but there's, they're not perfectly circular. They are white in color, and so all of those things are characteristics of colonies. So that's B. subtilis. We didn't test that one, but it's a good example. S. marcescens, you can see, actually has some pink colonies. So if I hold this up to something white, you can see under the name, you see those pink? That is because S. marcescens is actually pink. And so this is one of the things that we're going to be separating. So we know if it's pink, it's probably S. marcescens. And if it's white, it's going to be E. coli. So you can see that these colonies are much smaller than the B. subtilis. And so these solid media are really great for separating individual colonies, for testing for contamination, and for learning more about individual microorganisms. Whereas liquid media is really great for growing a whole bunch all at once if you want to send these off somewhere, or if you want to study them en masse. If you need a whole bunch of sample, you use liquid. If you want to learn more about what the colonies look like, you got to use plates. And if you want to figure out if you've isolated something, you have to use plates. And you can use the quadrant method, which we've done today, also known as the streak plate method. You can also do the pour plate method, where you would take individual tubes and you'd make dilutions here and then pour them into several plates of a solid agar, not of a broth. Okay, so when you come to lab, you are going to make your observations of the 1-to-1 mix, and then you're going to try to isolate it. So when you're isolating this, you're going to take a fresh plate, and then you're going to draw a line up the side, and you're going to label it E. coli. When we're labeling plates, we want to make sure to always label around the edge, because where we're looking is right in the middle. It will make it very difficult to see the individual colonies if you have to look behind your writing. So we always label along the edge, or even along this edge right here. So you put what it is, the date. Today is the 15th, 2019, and your initials, and then any other indicators you want. And on the other side, you're going to write S. marcescens. Which maybe I spelled right, maybe I spelled wrong. Okay, and then after you've got your quadrant streak, which will be grown by then, you take your loop, you demonstrate aseptic technique by making sure that this is nice and clean throughout, and then you're going to pick an individual colony from your quadrant plate. Just pick and say, oh, I think this one's E. coli, so I'm going to streak it on the E. coli side. Then you're going to flame it again in between each sample, even if it's of the same culture. We just want to minimize contamination. You're going to flame it all the way up the shaft again to make it all red hot. You're going to wait 20 seconds. You're going to pick the pink one, and then you're going to streak it on the S. marcescens side and say, I think that one was S. marcescens. Then we're going to see how well you did after we incubate these for 24 to 48 hours. Okay, and so that's how we're going to do the isolation of E. coli and S. marcescens from the 1-to-1 mix. You will be provided with the day 1 plate, and you're just going to make the observations of this pre-made plate. So that's why I made this video, so you'd see the origins of the plate, and hopefully you will contact me if you have any questions. Thank you so much for paying attention and for being prepared for lab.